Okay, everyone, we'll go ahead and get started. So thank you for joining us for this month's Science Cafe on neuroinflammation. I just want to take a brief moment to introduce the CTSC for those of you that are not familiar. So our goal is to improve the health of those in Northern Ohio and beyond by providing resources and services to our hospital and university partners, clinical and translational researchers and scientists, as well as community members. The CTSC is currently doing this by focusing on these 5 key areas and initiatives. So resources and services, workforce development, community engagement, health informatics, and clinical and translational funding opportunities. And I want to take a moment to introduce our presenter for today. So Dr. Zahoor Shah is a professor in the College of Pharmacy and Pharmaceutical Sciences, the Associate Dean for Graduate Education and Research, and the Program Director for the Department of Medicinal and Biological Chemistry at the University of Toledo. Dr. Shah's research interests are centered around the development of neuroprotective agents targeting neurodegeneration associated with stroke and cerebrovascular diseases. He has several patents related to this work. So I will turn it over to you, Dr. Shah.
All right. Thank you very much, Mackenzie. Can you hear me well, everybody?
Yes.
All right. Thank you very much. Good afternoon, and thank you very much for taking your time. So I'll get started since we have less time for presentation. So the topic neuroinflammation, it's a very interesting topic. And right now we know that neuroinflammation is increasingly recognized as a precursor or a driver of neurodegenerative diseases. And it is not just a symptom, but an active process that is believed to be preceding the actual clinical onset of Parkinson's disease and Alzheimer's disease way before kind of years or decades before the onset. So that's why the topic is very important. And whenever there is any injury or any, normally neuroinflammation is important because it protects our brain from infections and foreign bodies. Just try to kind of clear that from our brain. But whenever there's a kind of an infection or an injury, these microglial cells and astrocytes in the brain, they get activated, they release cytokines to eliminate pathogens or they help in tissue repair. But if the injury persists or the injury is very severe in case of stroke, both ischemic and hemorrhagic stroke, traumatic brain injury, and spinal cord injury, then these astrocytes and microglial cells, they secrete over too much of these cytokines, and that leads to the secondary brain injury. And in terms of neurodegeneration, we have seen that it precedes Alzheimer's disease, Parkinson's disease, and it has been observed in aged brains that these microglial cells are activated as compared to the younger brains. And then they also have a response towards autoimmune diseases due to persistent infection and microglial activation and astrocyte activation. So that's, in those cases like ALS and MS, multiple sclerosis. So it's a very important field, and we know that too many people are surviving for longer, kind of living more than expected times. And then we have more and more people coming with these neurodegenerative disease. And with stroke, incidence is going up every decade. So whenever there's a hemorrhagic stroke, as you see here in the on the left, or TBI, traumatic brain injury, with that the hemine, iron, which comes with the products of the blood, they activate these microglial cells. Once these microglial cells are activated, they release these toxic, normally as a protective mechanism, but too much of these mediators like TNF alpha, L1 beta, PGE2, nitric oxide, and peroxynitrite, all these H2O2, they activate, they lead to a direct neurotoxic signal. With that, and when the neurons, they start dying, with the dying neurons, they secrete their own signals like DAMPs, death-associated molecular patterns, ATP, nucleic acid, and high mobility group PROX1, which is a transcription factor, and neuromelanin. And with that, there's a perpetual microglial activation and reactivation, and that's what we have a primary injury after the stroke or the TBI and then secondary injury after that. So we are interested, and there's a conundrum about that. What should we target? And since my lab deals with drug discovery and translational research, so there's a conundrum, what should we target? And we have two phases of these microglial activation. We have the positive ones and the negative ones. The negative ones are initial injury, and then positive ones are the secondary injury, when you have all neurotrophic factors which are secreted by these microglial cells, like BDNF, brain derived neurotrophic factor, and then others, GDNF. So we don't want to, so this conundrum is that whether you want to block the good, the bad, and then let the good come really strong. So that's the conundrum there. We want to block the bad part, which is the primary injury. And in doing so, we also want to enhance the good part of these microglial cells. So the drug discovery should be focused on something like that to block the bad part and then enhance the good part. So that's our idea. And then we got involved when I started my lab in 2009 here at University of Toledo, transitioning from Johns Hopkins University to the University of Toledo. We got interested in this wonderful molecule, coughlin. Coughlin has been shown that it has a role in muscle cell contraction, cell movement, so many things, neurite outgrowth and neuritogenesis. But when there's an imbalance between phosphorylation with a regulatory mechanism of coughlin, it starts forming rods with actin filaments, and these rods accumulate in the synapse that can block the transmission. And it has been observed in Alzheimer's disease brains, human brains, that those rods can accumulate, they can stop the brain synaptic function, and that is one of the hallmarks also in Alzheimer's disease brains, in addition to beta amyloid and phosphorylated tau. So what happens whenever there's an injury, like ADP and glutamate is increased because of the injury, they activate NMDA receptors and P2X receptors. With that, calcium goes up, and then calcium going up can activate columodulin kinase and then calcineurins, and then they can activate slingshot phosphatases. When phosphatases are activated, they can dephosphorylate coughlin, and coughlin is converted into coughlin. That means active coughlin. And with low ATP in neurodegenerative diseases, they can also chronophin phosphatase activate them. With that, what happens that this coughlin can bind with beta actin. They can form dimers and trimers, and these actin filaments, they can accumulate in the synapse, and then with that accumulation, they can lead to synaptic dysfunction. And as I said, that it has been already shown that in autopsy Alzheimer's disease brains, there's a lot of accumulation of this. And as I said, that it precedes the clinical onset of Parkinson's disease and Alzheimer's disease. So we have also observed that this coughlin, once this is dephosphorylated, it can come from nucleus and then also cytoplasm. It can translocate to mitochondria, and that can release cytochrome C, and that can lead to apoptosis. In association with F-amyloid beta, it can activate FOC kinases, and then that can phosphorylate limb kinase and TES kinases. Again, there's a balance between coughlin phosphorylation, dephosphorylation, and if that balance is impaired, that can lead to, and then we have observed that can lead to microglial activation. And with that, there are very few labs in the United States who work in hemorrhagic stroke models. And I always talk to clinicians, and they always tell me that it's a very difficult disease to work with. There's nothing they can do. A patient suffers from hemorrhagic stroke, comes to the hospital. Most of these patients, they die. 50% of the patients die in the hospital. Only 50% patients who make out of the hospital, they suffer from severe disabilities, motor deficits, and also post-stroke cognitive impairments. So as you see here, is a big hematoma area. As you see, this is a midline shift. Also, there's a kind of swelling on the left side. So it's very hard. They just the idea is to get the patient in the hospital. They give just try to monitor blood pressure and bring the blood pressure down. That's the only treatment for hemorrhagic stroke. And there's nothing like for primary injury and secondary injury, as I showed you in my previous slide. So we tried to develop this model once I started here in 2009, and this is what we can do. We put the mice, this is stereotaxic stand. We inject collagenase as a chemical that can disturb the basal membrane of the blood vessels, and that leads to the breakage of those vessels, and the blood comes out. And we're trying to, this is a bregma here, this is a mouse brain. We go 2 mm lateral and then 3 mm ventral. We inject this collagenase in the mouse brains. And then as you see here, we try to simulate the conditions. You see that hematoma area in the striatum area of the mouse brain, as in it simulates what we see in human brains. And then if we are working on gene products, we can also give injections that can directly go in the area. And then you see that we can give injections. And then if we're working on gene therapy, we also have developed these neurobehavioral assessments, because we know that these hemorrhagic patients, they suffer from motor deficits, they have paralysis on one side. So we have developed these assessments where we can see whether their motor functions are improving with the gene treatment, gene therapy treatments, or any drug treatment. So as you see here, they stay on this rotor rod. If their motor functions are good, they will stay longer. But if they're not doing really great, they have the stroke or hemorrhagic stroke, they will fall down. And we can write down the timing, and we can assess the motor function with that. We can also look at their appearance. For if the mice is doing really great, as you see here, you get the 4 score. If they are doing really bad, as you see, this mouse is really doing bad, and we can give them severe score, which is 1. And then we can also look at their grip strength. And as we know that humans also suffer from paralysis, we can also look at their grip strength, and we can measure that. So we were interested in this coughlin protein. At that time, we didn't have any compound or a molecule developed that can target coughlin. So at that time, we relied on siRNA. So what we did first is we took the perihematoma tissues of this mouse brain, and then we looked at the coughlin expression and phosphocoughlin expression, and we found that coughlin is increased in this perihematoma area. So which gave us an idea that if it's increased in Alzheimer's disease brains, it's increased here also, it might have a role in this disease condition. So we developed this siRNA, and we gave directly into the brain. I just told you, as I mentioned, that we gave 3 injections in the striatum, and then we were able to see that we were able to see that at least more than 60% decrease in the gene in this area. So with that, we thought we should do an experiment. We should go directly into the brain, and then, but we decided that we're going to do 3 days before the hemorrhagic stroke. With that, so we were not sure initially what the half-life of this siRNA. So we gave 3 days before induced hemorrhagic stroke, and then survived these mice for 3 days. And we did all the recording, all the assessments, like neurobehavioral deficits. But we took the brains out, and then we found that hemorrhagic stroke, it was a big damage in scramble, which is kind of placebo. But our siRNA treated, coughlin siRNA treated animals had a lower hemorrhagic injury, and it was significantly different. And before killing the mice, we did behavioral assessments. We found that these mice were doing really great, appearance-wise, walking-wise, and that they stayed for longer time points on this rotor rod, and their grip strength also increased. With that, we also took the brains out. We did coughlin, as this is our primary target. We found that this placebo treated, they had a lot of coughlin activation, but with the siRNA treated, we were able to see that coughlin protein was down. And we also looked in the IBO1. This is antibody where you can localize under the microscope. You can localize this coughlin this microglial activation. So we're able to see that our drug, this siRNA treated animals, have lower activation of these microglial cells. With that, we also were fortunate to have Dr. Marak, Robert Marak, who moved to now Washington University. So he gave us these human autopsy brain samples of hemorrhagic patients, 63-year-old, PMI 24 hours within the death. We got these. They got these samples. We probed them with the same antibody for coughlin. We saw a lot of coughlin activation in the perihematoma area. We also were able to observe a lot of microglial activation in the perihematoma area. We took two samples. One was myocardial infarction, and the other one was rupture of abdominal aortic aneurysm. And then they were younger as compared to the 63-year-old. We observed that there was a little bit of coughlin, but not as much as we found in hemorrhagic stroke. But this gave us an indication that coughlin is also present in these cases we took as controls. There was a lot of microglial activation also, and that led us to think that this coughlin has a role, microglia activation, as I mentioned, that in aging also these microglia are activated, and since they have arthritis, peripheral systemic inflammation going on, that had led to their microglial activation also, and then coughlin is also increased in them. So that led to other projects where we're interested in seeing what happens to this protein in aging. So with that, we thought that now there's enough evidence of this role of this coughlin. Why don't we look at that, the coughlin protein, and start looking for grooves in that protein or the binding sites of this protein. And we were able to see a binding site, and we did an in silico study where we looked into thousands of molecules from the using that CIVS doc. We were able to see that one lead molecule was able to bind to this efficiently, then but not as efficiently as we wanted to. Then we took this lead molecule, we modified it, we kind of inserted bioisost here on the para position of this molecule. With that, we were able to see that this molecule binds to this protein and inactivates this protein efficiently. And then recently we did study, one of my collaborators at Stanford University, he was, so far it was an in silico, but we wanted to, reviewers always want you to do real ones also. So we did a kind of chip assay where we're able to show that our coughlin, the compound we identified, we patented also this one, was able to bind to the coughlin protein. So that was a good finding. And with that, we thought that we should do one initial experiment where we culture neurons. We added thrombin. Thrombin is one of the components of the hemorrhage. Whenever hemorrhage is, there is hemorrhage. Thrombin is one of the components. We took 10 nanomoles of this thrombin, we added to these neurons. We were able to see that there was a lot of cell death because of that neuronal death. But if we add coughlin inhibitor with the thrombin, we were able to restore the neuronal death. So that gave us another indication that our molecule is working in cell cultures. Let's move to in vivo studies. So at that point, we got this R1 grant to develop this drug therapy. So we thought that we should do one experiment. What we're gonna do, we're gonna induce the hemorrhage, and then after 3 hours of hemorrhage, we're gonna give IV injection of 25, 50, and 100 milligrams of this protein, of this compound, first after 3 hours, and then IP after every 12 hours for 3 days, and then have to do all rotor grip strength and T-maze. I'm going to explain what the T-maze is. T-maze is required. It assesses the working memory of these animals, because we know that after stroke, hemorrhagic stroke particularly, they suffer from post-stroke cognitive impairments. So with that T-maze, we're able to assess that too. So with that, as I said, that this is what happens. We were able to see that they stayed on rotor dose dependently with higher 100 milligram, they stay more. And then they had a better grip strength with the drug treatment. They as compared to the vehicle, and then they did really better on cognitive impairments. Their cognition improved. As you see here, these mice, you put on one arm of this T-maze, you put the cheese or anything, food, they will sniff and they will go to the side, left side. If their memory is intact, they will go, next time you put them again there, they will go to the right side. That means their memory is intact. But once their memory is impaired, they will keep going to the left side, and they will do these, they would never go to as their exploratory nature, they wouldn't go to the other side because their memory is impaired. But with the drug, we were able to see that these animals were making good choices, they were going to the right side. And we looked at the hematoma volume. Our drug was able to reduce the hematoma volume. And my collaborator at Stanford, he did MRI analysis on them. He survived the mice, same experiment, but he used lower dose, 10 milligram per kg. And then he survived these animals for 60 days. He was able to do MRI at 7 day, day 30, same animal. In our case, we have to kill the animals, different cohorts, but he can do the same experiment on the same animal. He was able to see that there was a less injury, hematoma volume at 7 day, 30 day, 60 day. One important observation he observed was that there was a rebleeding in these animals. So they had, and it's very common in humans also, they suffer from hemorrhage, and then after they suffer from rebleeds, and most of these people, they die of those rebleeds. So in these animals, he was able to see that the treatment not only prevented the initial brain injury, but also prevented these animals from rebleeds, which was very good observation. And he was able to see these are independently done at their place. We didn't have any influence on the experiments. And then he was able to see that coughlin is increased with the vehicle, but this coughlin inhibitor was inhibiting the coughlin, and that's where we see a lot of improvement. Then we got interested also in vascular dementia, which is totally different. You can, in animals, it happens in humans with atherosclerosis. In animals, what we can do is we can tie the common carotid artery, we can reduce the blood flow to the brain, and then we gave the drug immediately after this thing, because this is a chronic disease. We gave the drug for 3 weeks at 10 milligram per kg, 3 days a week, IP. And then we did, this is a Morris water maze. It's a different experiment setup where you have 4 quadrants. You put the mice, you do the habituation, you show them platform here. And if they're not able to find it, you can show them the first day. And then 4 days all training. And then the 5th day is the trial one. So as you see here, I'm going to turn it on. Wait a minute. So as you see this, the vehicle treated animal is doing circles and circles, and we have to do it only for 1 minute. While as the drug treated animal is able to find it within 11 seconds, and he finds the platform. But this vehicle treated animal is doing circles and circles, and even at the end, it takes more time. So we can do the assessments, and then we can draw the graphs, and we can see that our drug is doing better, and these animals are doing really great in terms of spatial memory. And then you have, with this 4 quadrants, you have kind of cues on the walls, so that helps these animals to find out what the platform is. So there's a lot of, it takes 5 days to do this assessment. And one time there was a company that was interested in my compound, and they, neurotrauma company, they were interested to take over, but then something changed in the management. Then they were no more interested. But anyway, I thought that I should do the studies and submit a grant for that. So this is how we do it. We take this kind of skull out and then hit the mice, and you can increase the severity. This is the moderate TBM model. We did the same experiment. We get 225 milligram per kg IV first injection, and then after 4 hours, and then daily after every 12 hours, we gave for 14 days. We were able to see that they stayed more on rotarod. They had better cognition, because we know that TBI patients, they also suffer from cognitive impairments, depression, and we were able to see that the depression levels went down. They were moving a lot as compared to the ones who got placebo. Their injury volume was lower as compared at 3 day and 14 days as compared to the placebo. And since we were looking at coughlin, we were able to see that the microglial activation was down, their coughlin levels were down. So this is unpublished data. So I'm trying to also develop this study. I have submitted grants on it. So far not been successful to get that funded, but this is another area we're interested in. Since we were able to see that our compound is enhancing cognition, so we thought that why not we try in Alzheimer's disease. And we know that coughlin these coughlin rods, they build up in Alzheimer's disease. That's another hallmark besides doing beta amyloid and then phosphorylated tau. Why not give this drug to these triple transgenic mice which develop this full-fledged Alzheimer's disease by 6 months? You can see early onset, but 9 months they have full-fledged Alzheimer's disease. So we took 9 month 11-month aged animals, both males and females. We took the brains out of these animals. We found that both the scaffolding was increased in their brains, overall brains. Then we did the same teammates. We gave the drug 3 times a week. At 7 day, we did the assessment. We found that their cognition was improved. We did the same thing. We continued this drug treatment for 2 months, and then again did the TBS. I mean their cognitive assessments. We found that these animals were doing great. Their cognition was improving as compared to the vehicle ones. So that was another study, and I was able to submit a grant on this also. It was not got a good score, not a score, but a good reaction from the reviewer. So we're going to submit that. So I have another collaborator. He works on this Angelman syndrome, and he has these mice. So we did that. We gave this same drug to them. It has the same these children. They have these developmental delays, intellectual disabilities, speech impairment, also problems with balance and coordination. So we gave this drug to those knockouts, and we were able to see that those animals stayed longer on the rotor rod as compared to the ones who didn't get this drug. So that's another, and these were done independently by my collaborator Julio at University of Texas Health Center, and another independent assessment of our drug that it is working in another model where you have a neuroinflammation and also problems with balance and coordination. So now where am I? So right now we did all the studies. We did in vitro efficacy, in vivo efficacy studies in TBI, hemorrhagic stroke, Alzheimer's disease. Now the next step is to do non-human primate studies, and I have a collaborator at Stanford. We were planning to do piglet studies in hemorrhagic models. We need more funds for that, and that's the difficult part, to move from these studies and go to the clinical trials, early stage clinical trials, phase 1 clinical trials. That's the difficult part. So we are stuck on this one. My auto one is on a no cost extension. All the money is done. We don't have more money to do the experiments. So we're looking forward to, I'm submitting grants actively, all those grants where I can take this drug to the clinical trial. So I'm going to explain that in my next slide, what those areas are. So my options are either I can license this drug to a company or venture capitalists. I had a meeting with a couple of venture capitalists. It's very hard to sell this drug, since the Alzheimer's disease, these drugs, they're going to cross the clinical trial, they don't go into clinic. Many drugs have kind of failed in clinical trials, so don't see really many people who are ready to buy this compound and take it into clinical trials or license it. So other idea is to try UG3 UH3, which is a mechanism where you can propose to do the studies, and then NIH will do phase 1 clinical trials. I submitted the grant in July, so hopefully I'm looking forward to that. If it gets funded, then the NIH will take over. They will do all the studies. The contract companies will do all the studies. We will do only the in vivo assessment studies, like the piglet model. That's what we have proposed in that. And then also I have another option, which is STTR, which is a small what is that called, the startup grants, not the startup grants. I can submit that. I can have my own company, which is the SBIR grant. I can submit from university also. I can have my own company and then do it. Those are my options. So if people or if scientists or researchers are working in this area, drug discovery, then the options for them are they can apply for U01. I submitted one U01 to develop this compound for Alzheimer's disease. As I said, that I got very good response from the reviewers. We're going to resubmit that, and then hopefully we can develop that drug for Alzheimer's disease, as we found that this is really improving their cognition in transgenic mice. The other one is I mentioned the UG3 UH3. It has two phases. Phase 1 is UG3 and UH3 is the second phase. where you can propose that UG3 do studies in your labs, UH3 and IH can take over. They can do studies. Their CROs can do studies on it, like PK/PD studies in bigger animals. They can also do phase 1 clinical trials if we get really good results with that. So I have submitted this one. It's a lot of learning curve, learning process to for a academician from a university to do drug discovery, and then was not easy, but it's a learning phase. I'm learning how to write these NIH UG3. So far, I have written R01 grants, but it's again too much to learn UO1. It's a totally different mechanism. But anyway, it's better not to write one. It's better to write one so that you have a chance that they get reviewed, and then you get the comments and you can improve on that. That's the idea here. Then, as I mentioned, that these are small business programs like SBIR and STTR grants. We can also submit those ones. So those researchers who are working these areas, they can target these kind of mechanisms. Uh, for those who are here and work in this area or have any molecule or any gene therapy and want to collaborate with us, so we have these models: neurological, neurodegenerative models, Alzheimer's disease models, stroke models, both ischemic stroke and hemorrhagic stroke. We have TBI models, where we have a repetitive model and single model, which is repetitive model is that for sports people who play football, they get hit, they are off the field for a week. That's what we're trying to do, a repetitive model. Hit the animals, leave them for one week, bring them again, hit them again 4 times, which kind of replicates what happens in sports. Or brain injury like single, we can go to DOD. That's what I'm planning to do. With the single model, we can kind of focus DOD funding. And then we have transgenic mice models. We can do that. We have in vitro models where we have these disease models, like we can expose these neurons to H2O2, heme, iron, oxygen glucose deprivation, so that can simulate the conditions of human diseases before we can take over to the animal models. And now, since we know that NIH is moving from these animal studies, there was a kind of a thought that they don't wanna fund wholly and solely animal studies. Now we have to develop human organoids. So we're planning to do that, since we have to keep up with all these changes, and we're trying to develop human organoids so that we can at least in our grants have one aim where we're going to try human organoids, not totally animal studies. So that's the one impediment right now, but anyway, we'll have to keep changing with the changing environment. So with that, all the acknowledgement, my postdocs who did a lot of studies on that, my PhD students, the SZ3 is based on my PhD student, Saleh. S is his name, S is Saleh, and Z is Zahoor. And then, as I said, that we patented that molecule. And then my collaborator at Stanford, Marco Rust, we got some coughlin knockout mice from Germany. Dr. Tilkaratne, he's a professor. He is a collaborator he developed this molecule, and he is also on our patent. And then Robert Marag helped us with these human autopsy brain samples. Isaac Schieffer is also, I have collaboration with him to develop drugs for Alzheimer's disease. So, and the funding from NIH, much appreciated. As I said that my R01 is almost ending, and we're looking for more funding, and without funding we cannot do anything. So these are very expensive experiments, and we need money to move. And also I've been funded by American Heart Association, so thankful to them for providing me money so that I can keep doing the research. I'll be more than happy to answer your questions. I took 5 minutes extra, sorry about that, but I'll be happy to answer the questions.
Great. Well, thank you so much for that presentation. There is a question in the chat, but I see Dr. McCumsey has her hand raised and it's her question. So if you want to unmute.
Yeah, hey Zahoor, actually, nice data. I'm not a neurologist, but I'm an infectious disease. So I am very interested in post-infections, like post-COVID, long COVID, and what happened in the brain. As you know, a lot of people have brain fog, other brain symptoms. So we were looking for a biomarker to try to see if there's anything that we can use to diagnose brain or neurologic long COVID. And we started talking about doing tau and NFL. How does cofilin as a biomarker in Alzheimer's disease, for example, compared to tau? Do you know?
So we see a lot of amyloid beta and tau in Alzheimer's disease patients, but this has been overlooked. I haven't seen anybody working on this coughlin actin rods. As I said, that neuroinflammation precedes the clinical onset. There's no drug, no preclinical study that has tried to target it. Now, coming back to the COVID fog and neuroinflammation pertaining to inflammation or the infection. So we're trying to model it. I think we published that study where we're going to give LPS, systemic inflammation, and we see that with aging that blood-brain barrier becomes weaker and the infection can go easily into the brain, and you can see that young animals they can prevent it from this LPS infection going into the brain, but aged animals, they are prone to this infection. We have seen that they have more of this A beta and tau and in our animal models, and also the microglial activation and coughlin activation. So that might be, I'm always looking for these things. That was a very good point. You figured out that we might be interested, it might be interesting to see in these patients whether they have kind of this coughlin activation in their blood, because there's a peripheral inflammation and all. If you are able to see that in CSF fluids, that there's a coughlin activation, coughlin is more in them. We try to see that in animals, but it will be really great to see if these patients have this kind of active coughlin, more coughlin as compared to the normal ones. Well, it might be a good field to go forward.
All right. So as the Science Cafe is to create new collaborations, so I will be in touch with you actually.
Very good.
We have patients, we have samples, we have funding also to be able to get those assays done. So I'll be in touch. This is one area, long COVID, because it's a chronic illness. This administration actually, as much as they're not as much anti-COVID, acute COVID, but they recognize that long COVID is a big problem for the US and it's costing a lot of money. So that is one area, and I'm just very interested in.
And there are studies showing that these COVID patients who had COVID, they are more prone to getting stroke and more prone to getting heart attacks. So it might be a good idea to check it.
Yeah. And a hint that we may all actually, since most of us had COVID, be at a higher risk for early cognitive dysfunction. So we have a country with, or a world with a lot of early dementia. Yeah, pretty scary.
Yeah.
Oh, thank you. That's interesting.
Thank you.
And we do have 2 other questions in the chat. I don't know, Roberto, do you want to unmute and ask your question? Otherwise, I can read it.
Yeah, I can answer. Are you able to hear me?
Yeah.
Perfect. No, this is a great talk.
Thank you.
So my thoughts are, one is, you talked about in the presentation about low inflammation being positive in the brain, but what are your thoughts on the cases where transient low inflammation can be a negative, especially when it comes to developmental stages of the brain? So what are your thoughts on these low transient inflammation and how that could affect the development of specific subtypes of neurons or, oligodendrocyte damage? And, this is also thinking, kind of sharing the thought with Dr. McComsey on, thinking of this in a point of view of a neuroviral infection like SARS-CoV-2. What about HSV-1 when you have persistent reactivation of the virus, and let's say it's think of it as an asymptomatic reactivation of HSV-1 in the brain. What could the landscape look like when it comes to low inflammation? And also, have you looked into brain-associated macrophages and whether or not they could play a role in picking up the slack of any microglia that have been, that are, let's say, there have been loss of microglia or such. And lastly, brain organoids. Now that we're in the, the current.
Yes, I got you. Hello?
Yes.
Okay, all right, so 3 questions. So let me try to answer all 3 questions. So chronic inflammation, as you mentioned, in infections. So neuroinflammation, if it's subchronic, it's microglial activation is required whenever there's a foreign body or any infection in the brain. They are required. But if there's a sustained, even a chronic inflammation, we have seen that aged brains who had chronic inflammation, which kind of because of arthritis, rheumatoid arthritis, their microglia are way activated as compared to the ones who didn't have kind of an infection. So that kind of answers for one question. The second question was, so in COVID cases also, you have this chronic inflammation going on. Chronic inflammation can lead to, with the age, we have this blood-brain impairment, and then the peripheral inflammation can cross these cytokines and peripheral cytokines and the chemokines. They can cross in the blood, in the brain, and they can activate these microglial cells. With that, in aging, also macrophages, they can also go there. They can also activate microglial cells. It happens with aging or injury also. If there's a breakage in terms of stroke, there's a breakage of blood-brain barrier, and then these macrophages have been seen that they can infiltrate in the brain tissues. They can activate microglial cells. Coming to the third question, organoids. Again, I don't know. There's so much of focus on organoids, and they're not cheap. Even developing organoids takes a lot of money, $5,000 one experiment, and they're not foolproof. Also, I mean, if you develop one organoid, they have their own environment, and in terms of drug development discovery, you have problems with that. Whether the drug is going to enter in because it has its own environment, it's going to diffuse equally within that organoid or not. So there are so many things with that, and it's not cheap either. So that was when this thing came out. FDA gave a presentation on it. There was a kind of survey about your thoughts. What do you think about it? So we're all we highlight that animal research is also important. Whatever you see in organoids, you again have to replicate that in animals before you can go to humans. So so that's a thing there, and we have to obviously we have to do it. We've been told by NIH to focus on. We've been doing that. We were told to do males and females. We did that. Every change we had to kind of do that. We have to write in our grants. So we have to keep going, and we have to probably try to do organoids also. I hope that answered all 3 questions. If I missed anything, please let me know.
No, that was great. That was great. Great talk. I appreciate it.
Thank you very much. Thanks a lot.
There is another question in the chat. So what are the chances if someone wanted to work on this research area in a lab outside the US? I don't know.
Outside the US, I don't know much about that, but it's easier to do these studies in China, in India, although there are regulatory kind of rules and regulations on that. But again, there are companies. It's easy to do outsource these studies to companies in China and India. So I've been told by companies I've been reaching out to do studies for us, PK/PD studies in bigger animals. So they have these companies either in Europe or India and China where they do these studies, and then that's cheaper than doing these studies here in the United States. I hope that answers the question.
I have one more question if time permits. So spatial transcriptomics, are any thoughts on following and performing any spatial transcriptomics on these brains now that there are, better techniques that's developed for better resolution? What are your thoughts on that?
I agree. So that was my comment on the grant to do more developing on molecular mechanism. We know that this is working, it is improving our memory and all that stuff. What is the mechanism? So I'm trying to team up with a collaborator here at UT, Robert McCallum-Smith. He has these omics. He can look into these kinases. So I'm trying to team up with him, do the kinase assays and look for further mechanism, which kinases our drug is inhibiting, which one is improving. The idea is like coming back to your 3 questions, the chronic inflammation and whether our drug is going to inhibit that, which compounds or the molecular mechanism, which kinases are it inhibiting, whether those kinases have a role in chronic inflammation or early inflammation or later inflammation. And then also do some RNA-seq. We did some studies with RNA-seq, but with the lack of funds, we were not able to do more. We did only 3 animals, but we didn't get any concrete kind of data from that. But with the collaboration with Dr. Smith at UT Neuroscience Department, we might be able to look into the kinases, at least the kinome areas. We can look into the mechanism and see what kinases our drug is inhibiting, or what kinases should be inhibited and not be inhibited. Again, the good and bad.
No, that's great. And I would also suggest, because I know that visual spatial transcriptomics is very expensive as well. You could also probably try to take a swing at bulk RNA-seq CyberSort.
Mm-hmm.
Essentially, which would kind of be a cheaper way of doing single cell.
Okay. Yeah,
Yeah, because that way it essentially matches your data against a database, and it extrapolates what kind of cell types you have and what types of changes in pathways. It's not as rigorous. It is not as rigorous as single cell, but it definitely will give you an idea and could be helpful for you as you develop your proposal.
Wonderful. Thank you very much for the suggestion. Thanks a lot. We'll keep that in mind. Yeah, we need money.
Okay, does anyone have any other questions? Feel free to unmute. I'm not seeing anything in the chat right now. I see Roberto put the CyberSort link into the chat for you,
Okay.
Dr. Shah. And
Thank
then
you.
someone had said, thank you for the great presentation.
Thank you, everyone, for being here.
I'm not seeing anyone with their hand raised. A lot of, yeah, nice talk and praise in the chat. I will just share this last slide then. So I'm just throwing up a few QR codes. Oh, there. So the one on the left is for the registration page. So if you want to sign up for either next month's Science Cafe on irritability and depression in cancer patients, or any future sessions in the series, you can access that from that QR code. And then the right QR code is to become a member of the CTSC. So you can be sure to stay up to date with all of our events. So thank you so much, Dr. Shah. That was a great presentation. If there are any last questions, feel free to put those in the chat or unmute.
Thank you, everybody, for taking your time and attending this talk. Thanks. I appreciate that.
Great. Thank you so much. Everyone have a great day.